Hello, listeners. Welcome to IT Guy Who Lifts podcast. I'm your host, Palash Gupta, Diploma in Nutrition and Fitness Sciences from INFS India, and Bachelor's in Sports and Exercise Science from Loughborough University, UK. The goal of this podcast is to understand science and science based tools with the help of industry experts so that we can further optimize our training and nutrition program. With that in mind, let's jump right into today's podcast. Hello, listeners. Today we have been graced by the presence of Dr. Gabriel Fundaro. So, without further ado, let me pass the baton towards Dr. Gabriel so that she can give the listeners a brief introduction about herself, her professional endeavors, her lifting endeavors, and how this iron bug bit her and made her the woman she is today. But before that, Dr. Gabriel, on this podcast, I always give this disclaimer. So, on this podcast, we actually urge and request our guests to provide the longest possible introduction they have ever given elsewhere. Because we want to know all the events which unfolded in front of our guests and laid down the path on which they have walked upon and become the person that they are today. So, with that in mind, stage is all yours, Dr. Gabriel.
Oh, thank you. Well, I'm glad to know that because I do have sort of a strange story. um, I had sort of like an unconventional path to where I am today, and I actually started college as a music major. So I thought that I was going to be a music therapist. I had never I never thought that I was strong in the hard sciences, and I didn't really receive helpful messaging about it when I was growing up. It was sort of like a stereotypical, women in our family aren't good at math and science, and so you probably won't be either. And so I had that belief, and I was pretty prolific in music growing up. I sang for my entire life. And when I went to college, I thought, okay, I'm going to just do music for the rest of my life. But as it turns out, I was skilled in singing and no other instruments. And as a music therapist, they kind of expect, understandably, that you will play a variety of instruments. So I had been accepted on a full scholarship for undergrad, and that required maintenance of a high GPA. And I sort of predicted that I might not maintain that GPA if I were to stay, as a music major. And in the meantime, I had really fallen in love with my introductory biology class. And I also had started getting into just exercise and rock climbing. We had an indoor rock wall at my undergrad institution, which is out in southwestern Virginia, so like the East Coast of the US. And I thought, well, maybe I can do something that's sort of like tangential to this. And instead of a music therapy major, I'll be a recreational therapy major. And the focus of that degree was about helping people learn about themselves and connect with others through like outdoor activities. But part of the requirements for that degree were to do leave no trace training. So you had to go live in the woods for like 2 weeks. And I thought, at that time, I wasn't as much of a fan of hiking as I am now. So that wasn't really for me. And then I thought, well, is there something that I can do that's like the combination of biology and exercise? And lo and behold, exercise science is a thing. So I switched my major to exercise science. And so the major was technically exercise, sport, and health education. And we even had the opportunity to do a minor in martial arts. So I actually took a lot of martial arts courses, but I ended up doing sort of a combined degree. It was a cognate. It was like commercial fitness and exercise science. So I thought that I was going to own a gym. So that was my plan until my junior year of undergrad. And I realized I was not interested in the business and marketing and liability side of things. I really was just passionate about the physiology of exercise and particularly skeletal muscle physiology. So at this point, I was taking an anatomy and physiology course that was really It was expedited because I had switched majors, so I was a little bit behind. I got really lucky that I tested out of a whole year of English, but I didn't really have time to do the full 2 semester anatomy and phys. So I did the equivalent of 2 semesters in one. So it was like an 8 credit course or something crazy like that. Extremely quick pace and really frequent labs, and I loved it, and I seemed to really take to the information, and I ended up tutoring my classmates entirely on accident. I just showed up to one of our study sessions one day, and they handed me a whiteboard and dry erase markers and said, we have so many questions for you. So I ended up tutoring, just kind of like, it wasn't official tutoring, so I was like, black market tutoring. And at the end of the semester, I confessed to my professor because there was like talk about cheating and things like that. And I thought, oh my gosh, I hope I haven't like broken any rules. But he was really, he was pleasantly surprised, and he said, clearly I had been helpful because the grades had been really high that semester. And so he invited me to teach his last lecture on renal physiology. So I taught myself the chapter, I worked with him, I developed a lecture, and I taught it. And that was when I knew that I wanted to go on to become a professor. I thought, if this is something that I'll do for free and I love it, then I want to do this for a living. So I went straight from bachelor's to PhD. And I interned before I started my PhD as part of my bachelor's in a skeletal muscle physiology and biochem lab. And that was where I ended up doing my PhD. I basically just transitioned in after my semester of internship, and then a semester as just a research assistant. I applied for the PhD program and was accepted. And I started in that program studying the effects of high fat feeding on skeletal muscle hypertrophy. That was my intended project. And part of my duties were I were doing a lot of like animal husbandry. So I was handling the mice, I was giving injections, I was sacrificing them, and I was frequently performing these LPS or lipopolysaccharide injections. So we would inject the mice, and then some 6 hours later, we would sacrifice them, collect their tissues, and then look at markers of inflammation and measure metabolic flexibility. And I was really curious as to what this was modeling in a human body. What's the physiological relevance of LPS injection? And that's when I learned for the first time about the gut microbiome. I literally did not know that it was a thing before this. And this was back in 2009. So it really wasn't something that was being spoken about frequently. And and so my advisor just kind of gave me a brief overview that LPS comes from certain bacteria that reside in our intestines, and there are certain states like type 2 diabetes or obesity or high fat feeding that are associated with increased levels of this LPS, and that has been cited as a potential cause or player in the development of type 2 diabetes and sort of low grade inflammation that they termed metabolic endotoxemia. So I'm very much sort of like a problem solving, big picture puzzle type of person. And I thought, why are we not looking at the level of the gut microbiome and trying to stage some sort of intervention there instead of, just treating at the level of skeletal muscle sort of after this has occurred? And we just didn't have the funding, and we didn't have, and that wasn't really our area, that no one in the lab had ever done anything with the actual gut. So I was curious and wasn't able to quell my curiosity for about 6 months until we actually procured funding from a company that was producing a probiotic called VSL3. And once we gained that, when we were given that grant, my advisor came to me and said, "Do you want to do this as your side project? A little like probiotics thing on the side." And I was like, "Oh yeah, great! This is so cool! I've got my main project going off without a hitch, which never happens. There's always some sort of mistake, but it was so smooth." And then I got the side project, and I'm like pursuing my curiosities. So fast forward another 6 months or so, my main project has reached the tissue collection stage. So we're, sacrificing all these mice, collecting their tissues and putting them in a storage solution. And we had some undergraduate, helpers and whatnot that were assisting with the process. And I was not, keeping close tabs. And we ended up improperly storing the pretty much all of the skeletal muscle from that study and lost all of it. And so we had to can the whole thing. There was literally nothing that we could do with it. So fortunately, I had this side project probiotic study that we just said, okay, let's make that the topic of my dissertation. And so that's how I ended up studying the gut microbiome and probiotic supplementation. And that took 5 years, in part because I had skipped my master's, and then also because I started a teaching fellowship in my third year, and it was intended to be a 3-year program, but they kind of gave me a pass as long as I got all the requirements done in 2 years that I could finish and graduate with that. So I went on to become an assistant professor of exercise science, and I didn't intend to do anything with my dissertation research because, again, this was in 2014. So like the gut microbiome, like gut health was kind of just emerging, but people still weren't really talking about it, to any great extent. Like the internet was still focused on just like inflammation, so they weren't talking
Yeah.
about gut microbiome. And I taught exercise science and sport nutrition for 3 years. And in my third year, I was, I guess, discovered by Mike Israetel from Renaissance Periodization in a Facebook group associated with the International Society of Sport Nutrition because I was studying for that exam. And he liked the content that I was putting out. I had like a tiny little, sport nutrition blog, and he, recruited me to RP. So I was an RP coach, and I did that while teaching. And then I started my own telehealth business, Vitamin PhD Nutrition, all within the same year. And at that point, I'd been teaching for 4 years, and I needed to either go up for promotion to associate professor, or kind of like decide to not stay in academia. I mean, you kind of have to like, it's like you go forward or you leave. So I was not feeling as fulfilled in my academic role because it wasn't kind of what I had anticipated it was going to be. And I was feeling very fulfilled by coaching. And I had done a couple of speaking engagements and found those to be incredibly enjoyable and fulfilling. And I made the decision to resign from academia and go into coaching full time. And from there was just kind of a snowball effect because Mike had set me up with Steve Hall from Revive Stronger.
Sure.
Yeah, for my first podcast. And I talked about like just the basics of gut health. And I think that might have been one of like the first podcasts where people, were kind of talking about that. This was in 2017. And from there, I, started speaking internationally with RP. And I spent 4 years and some change with them while I was building my own business. And then I started collaborating with my friend Shannon Beer on our Bridging the Gap article series that talks about sort of the history and sociocultural influences on dieting and sort of diet culture. And we developed the comprehensive coaching framework that operationalizes motivational interviewing, acceptance and commitment training, and cognitive behavioral coaching in a framework that helps to increase client capacity and focus on flourishing health rather than just physical health or just aesthetics. And we've done several webinars together, and we've also developed a spectrum of intentional eating. So it is a sort of this anti-dogmatic, nutritionally agnostic spectrum of weight neutral and weight focused approaches that's integrated into our coaching framework. So we're helping people determine what is the best approach for them. And so in the past year, I've separated from RP. I did finish my book with Dr. Hoffman, and we published that through RP. And then I started up with Examine a couple months later. And I've been a researcher and writer for them, focusing on gut health related topics. Yeah, so that's kind of where I am today. So I got here through just like a couple happy accidents and, kind of being in the right place at the right time.
Oof. So all the serendipitous events that happened finally led you here. So that was really a great introduction. And you not only tested the waters in the lifting side, but you went skin deep as well. So would you like to throw some light in that? Because uh, people will are really interested in knowing your lifting background as well.
Oh, sure. Yeah, so I started lifting when I was 19. I made friends with someone I think he was in my anthropology class. His name was, well, we would call each other by our last name, so Wisely. And he was sort of like a curious, interesting, like introverted character. And I can't remember how we started talking, but we ended up like either hiking or going to the gym together or something one day. Then I wanted to get stronger for rock climbing. And so I didn't know what I was doing. I hadn't actually switched majors yet. And I just thought like, okay, I'm going to do a lot of like back exercises. Gosh, this is really fun. Like I want to learn more about this because I'm the type of person that like wants to learn everything about a specific topic.
Yeah.
So when I finally switched to exercise science, that was his major. We were in a super small program. So we had pretty much all of our courses together, and we were like the two of the biggest gym rats. Like people in our major like knew that we were going to be at the gym or out hiking or whatever. Playing tennis. It was just like I'd never been active for my entire life. I'd never really like I played, basketball in sixth grade, and that was about it. So I was like, oh my gosh, like there's this whole world of like fun activities, and lifting. And I ended up doing a bench press competition, I think like my junior year of undergrad. And I was like, man, this is amazing. I feel just so empowered. So I kind of dabbled in distance training until I got into my major, and then I was able to really, like produce an okay lifting program. And I've maintained that. I mean, I have never stopped lifting since I was 19. But I've dabbled a little bit in trail running. So I lived in the mountains of southwestern Virginia for 10 years, right next to the Appalachian Trail. So like I would modify my lifting program to fit with the amount of running that I wanted to do. So I was out running for, 7 to 10 miles on the trails. And I ended up injuring my ankle so severely that I couldn't keep that up. And so I kind of just like went back to lifting. And then once I felt better and I moved to Georgia to start teaching, I was like, oh, I've got like disposable income and a little bit of free time now. And so I started Jiu Jitsu. I did that for about 9 months. And then I did a physique competition. That was my one and only. And I was like, yeah, okay, that was nice. But I don't want to basically give myself an eating disorder again.
Yeah.
So, I was like, I'm going to take a break from the long-term, starvation. And I turned my attention back to, gaining strength and lifting. And it's when I got into powerlifting. And I did several powerlifting competitions. I must have done that for a couple of years. And, I ranged from like not placing at all. There's one meet that I got like best female lifter. They're really small meets, but, it was just kind of a neat journey to focus on strength and kind of like also change my perspectives about like about body weight and weight and like focusing on strength rather than trying to be like my smallest self, which is sort of this pandemic of a lot of women in the US. So
Yeah.
after my venture into powerlifting, I kind of took a break from that because I was competing pretty much back to back and I was starting to get a little bit tired. And I just thought, maybe I'm going to chill out and like, exercise more recreationally. And not soon after that, I started traveling with RP. And then it was like, how am I going to fit lifting in with, traveling around the world? And so I had to be more flexible with my training program, and that was difficult for me because I'm usually a very like structured and organized person. And then and then I started just living as a digital nomad, so it was like I'd maybe go to like an Anytime Fitness or whatever. And but I still kept it up because I just really intrinsically am motivated. I just really love to lift. And then when the pandemic hit and all the gyms closed down, I think a lot of people who are like lifetime lifters, you had this crisis of identity. Like, what do I do
Yep.
now? Who am I if I'm not going to the gym? So it was a lot of like resistance band work, and I got a little like set of adjustable dumbbells and focused more on just being outside. I had moved to Arizona, and I just love to be out in the mountains in the desert. So I did that, and then I live with my boyfriend, and we have an in-home gym, which is very well equipped, and it's really convenient, because it's like literally outside the door of my office. So I that. And oh yeah, and I forgot, I started learning how to play ice hockey a couple of months ago. That's a sport that I've been wanting to learn since I was little because I grew up in Michigan and hockey's very big there. So my training more recently has been focused on hockey specific stuff, sort of like dryland training. And it's fun for me to do programming and like look at different sports and, what are like the
Yeah.
movements, that are sport specific. So, kind of keeps things fresh and interesting. And yeah, so that's what I'm doing now is like training like kind of a hockey player.
That's so wonderful. So, along with your professional endeavors and lifting endeavors, have you ever considered of giving audition for Wonder Woman? Because that title definitely suits you, based on your history of your academic endeavors and your lifting endeavors. So, you should get in touch with the DC Cinematic Universe and you should try for the Wonder Woman audition.
Thank you, thank you.
So one more question I have because I became really, really curious about the social media handle which people use, right? Just you have vitamin PhD nutrition. So what is the how this particular term coined up in front of you, or why you choose to decide with this kind of a term? Because our social media entity somehow becomes our second identity in the real life, right? So how you came about that particular term? Would you like to throw some light?
Yeah, absolutely. So I came up with that when I first started teaching, and I had my sport nutrition blog, and that would have been probably, I think that might have been like 2016 or 2015. So it was like not long after I started teaching. And my first sort of catchphrase was something like, get your daily dose of evidence-based nutrition info. So I
Nice.
wanted to, kind of like send this because I was talking about supplementation a lot. I was kind of like, that was my little like nerdy path for a while. Like, every sort of supplement, I wanted to learn all about it. So I was thinking like, supplements, vitamins. And then I thought like, I want to not to toot my own horn or be elitist, but to say like I have an advanced degree, and so I'm not just like making stuff up. I'm not just like an influencer, if that term even existed back then. But to say like this is an evidence-based set, this is an evidence-based blog. So it was like vitamin PhD, and and then I used to have parentheses around the pH, so it was like vitamin D, but vitamin
Uh,
PhD, but I sort of phased that out over time, in part because you can't really like use it's hard to do that, and make it look good. And I think my LLC is registered with just like the PhD. So that's where that came from. And over time, my content and messaging have changed so much that it wasn't that it started to become about more than just nutrition information. So as my messaging transitioned and I was speaking more about like the psychology of the change in weight loss, and I still am talking about gut health, but I'd say it's probably a 50-50 split. I realized that that doesn't like fit so much in terms of that sort of little phrase. So I still have vitamin PhD because at this point, like now that's an established
yeah,
name, I guess. Not, and again, it's like, as established as I am compared to other people is not established, but it's like people know that. So I kept that, but this year I changed the phrasing, and now I was inspired by a podcast I had earlier this year where the host was just very kind and gave me such nice feedback and said that I have this mix of both science and compassion, that I'm trying to communicate science, but I also want to validate people's experiences. And I thought, oh my gosh, I really like that. That's so kind. And like, yes, that is what I want people to get from my content. And so that's where I came up with science fortified with compassion. So I still have like fortified, kind of like related to vitamins and minerals,
yeah,
but science fortified with compassion. So it's science, but it's provided in a way that is intended to be kind, compassionate, and helpful.
definitely, definitely. Really love to hear the side story and the backstory behind that particular Instagram handle and your LLC name tag. And I read your book, I have read your blogs, posts, and the things which I really like the most is the way you portray these scientific jargons. And because we, as a science mediator, it's our job to portray the scientific knowledge into the actual application so that even a layman or a general population can use that particular scientific knowledge, and they can enhance their day-to-day life. So I definitely love that part of that. So I have read your book, but since this word gut health has been thrown around a lot, people do not completely understand it. They think it's just the intestine, what happens in intestine, rest in intestine, just like what happens in Corsica, rest in Corsica. So, would you like to throw some light? What actually is gut health, and what are the components that it circumcised by?
Absolutely. So I've developed sort of this working definition of gut health because I noticed the same thing, that it is this really ambiguous term that
Yeah.
people use it to mean all sorts of things. And in the research world, there's really not a consensus definition, and it's not used as frequently. So there are some things that are used in both places, like you'll see leaky gut in a publication, and you'll see leaky gut on an Instagram post, but you won't really see gut health in a publication. So I came up with this sort of working definition that is not derived directly from the research, but it's something that I think is helpful to get a sense of this like umbrella term and what people are probably talking or thinking about when they say gut health. And I call it the 3 Ds of gut health. It's diversity, disease, and digestion. So diversity refers to the richness, so like the number of species and the relative abundance of species of microbes in the gut. Or we can think of it in some types of microbes, they're not classified as sort of species, but sort of the variety of microbes in your gut, and then also the variety of genes. So there's a vast number of genes and just genetic information in the gut that greatly outweighs our own genetic information by about a magnitude of 10. So we have about 22,000 genes, and they have about 220,000. So it's, they have an abundance of functions. So ideally, we have a wide variety of microbes that are not closely related. We have a lot of redundancy built in. So if we lose some microbes, we don't lose the functionality of the whole population, and that we have a relative abundance. When you're saying relative, we're thinking about it like a pie chart. So if we have, 60% of one and 40% of another, now if we lose some, we'll have a 50-50 split. Or, if we gain some, we might have a 70-30 split, but it has to add up to 100%. So there's relative abundance of microbes that favors either the neutral or beneficial microbes and suppresses the virulence of potential pathogens. So we do have pathogens that are normal inhabitants of the gut, but in healthy individuals, they're just sort of existing. They're not actually causing illness. So that's diversity. And then disease. So disease is looking not at the microbiome, not at the microbes in their genetic material, but at the anatomy and physiology of the tissues of our gastrointestinal tract. So our intestines are made of multiple layers. So like the outermost layer, if you were to open up someone's stomach and you look, you see the outside of intestine. That's multiple layers of smooth muscle that are contracting to move food along. And then inside of that is a thick layer of connective tissue. So there's a lot of capillaries in there, so there's a lot of blood flow. There are tons of immune cells. And then on the innermost layer of the intestines, that's where we have this single layer of intestinal cells. So they are either helping to absorb our nutrients from our food, or they're producing mucus, or they're playing some sort of immune function. And then covering those cells in the small intestine, we have an incomplete layer of mucus. And in the large intestine, we have a bilayer, a double layer of mucus. And the top layer of mucus, the one that's like inside the innermost layer, that's inhabited by microbes. So when we're looking at disease, we can either look at a functional disease. So the tissues look normal, but they're not behaving normally. So there's some issue with the physiology, like with irritable bowel syndrome, or an organic disease where the tissues are being affected, like an ulcerative colitis. And we're also going to see a change in the function as well. So we don't have any causative relationship. We don't have a cause and effect relationship between the microbiome and any disease, but we tend to see patterns in people with
diseases, that their microbiomes look different from people without that disease. But even between two people with the same disease, we'll see a lot of variability. So we
Yeah.
can't establish cause and effect, and that's why the microbiome is really a separate entity from the gastrointestinal tract and the disease that might be present there. But people are usually, they're thinking about like the
Yeah.
microbiome and the tissues as well. And then the third D is digestion, and that's really just the subjective experience of digesting and absorbing our nutrients. So people are usually thinking about gas, bloating, stool quality, bowel movement frequency. And oftentimes people are assuming that if they have some GI discomfort, like, or having some gas or bloating, that there must be some dysfunction at the level of microbiome, diversity. They have like a dysbiosis, imbalanced microbiome, or maybe they have some sort of disease. And that can be the case. So irritable bowel syndrome is, a lot of the symptoms are just, it's GI distress, gas and bloating and, constipation or diarrhea. But some level of gas and bloating are just a normal phenomenon that happen because those microbes are fermenting nutrients and creating gas. And some nutrients, some specific types of carbohydrates are very fermentable, and so they create a lot of gas. And some GI diseases don't have, especially when we're looking at like colorectal cancer, that doesn't have like significant outward symptoms right off the bat. If someone has like a very small tumor, they might not know that anything's wrong, or if maybe like their bowel movements look a little bit more narrow, but they don't have any other issues, they might not notice that. So that's why it's important that, we are communicating the science with transparency and clarity, so people know that, okay, they probably don't have like a candida overgrowth, because that's something we only see in premature babies and people who are immunocompromised. So if they're having some real changes to their bowel movements and whatnot, and they go to like do a candida cleanse, but they really have a serious disease going on, that could cause significant harm. That's why it's important, one of my missions to try and clarify this, to say, might not be anything to worry about. So don't let someone sell you a problem that doesn't exist.
Yeah.
But, if something is going on, go and get, a valid diagnostic or screening test so that you can.
Definitely. Since you mentioned the three Ds of gut health, like diversity, disease, and digestion, and we know that digestion starts from our mouth and ends at the rectum. So this word gut, it comprises of how many organs, or you can say body parts? Would you like to throw some light on that?
Sure, yeah. So, and that's a great segue too, to mention that we actually have multiple microbiomes. So we have
Yeah.
an oral microbiome that has much lower numbers and much lower diversity. And that's where we actually could see, even in a healthy person, we could see a candida or a yeast overgrowth in the mouth, because it's easy for them to form biofilms. We don't have these sort of like fluid, like planktonic forms of the microbiome. We have kind of just like surfaces. So in babies, they develop thrush. So that's a yeast overgrowth. So that can occur in the oral microbiome. We also have a skin microbiome. So again, that looks all these microbiomes are significantly different. So if you were to culture them, a different set of microbes would grow. But we have a skin microbiome as well. Again, low numbers and lower diversity than what we would see in the gut. And then women have a vaginal microbiome. Again, much lower numbers and lower diversity. It is really predominantly inhabited by lactobacilli species, because they can withstand the acidic environment. And that is another place where females could experience a yeast overgrowth, again, because we have low diversity, and because the pH can be affected if a woman were to use like a douching product or something like that throws off the pH. Now we have less suppression of the yeast species, and so they could overgrow as well. But when we look at the gut microbiome, what we're really looking at, we have a gastric microbiome, so that's just the stomach. And then the gut microbiome is generally referring to just the small and large intestine. And we do actually have some other microbiomes that aren't as well characterized, but in the gallbladder, there's a set of microbes. And then in milk ducts, there are also microbes. So we could think about that as sort of a microbiome. And microbiome just means microbes in their genetic material. So it doesn't have to be, a super complex system, but we see that we have these microbes pretty much, in every area of our body. So that's kind of how we can break that all down. And then the fecal microbiome, the one that we're usually using in research, is significantly different from the gut microbiome. So when we look at the sort of the ecosystem of the small and large intestines, we can break that down into small micro environments because we have the lumen of the intestine, so that's the inner tube, and that's where the microbes are existing in sort of this planktonic state. So they're floating around in the chyme and the foodstuffs and the fluid that are present there. And the small intestine is much more acidic because it's close to the stomach, whereas the large intestine, especially the distal large intestine, is much less acidic, and it's also more anaerobic, so there's much lower oxygen availability. So both pH level and oxygen availability are strong influences on which microbes will be present. So if we have lower acid and less oxygen, that's why we have such a high number and high variety of microbes in the large intestine versus the small intestine. Then we can look at the mucus layer. So remember, in the small intestine, we have a very thin, incomplete mucus layer, but in the large intestine, we have that bilayer. So there are also mucus-associated microbes that are actually residing in the mucus layer. And when we take a fecal sample, where that fecal sample is more representative of the microbes in the lumen of the distal large intestine, and that's significantly different from the samples that we might take from the actual mucosal layer. So it's something else to keep in mind, when we're looking at studies, that unless we're taking a sample from, and some studies do, unless we're taking a sample from the small intestine and the large intestine and looking at the mucosal, inhabitants, that really the term we need to be using is the fecal microbiome rather than the gut microbiome.
Definitely. This piece of information is really, really important for the listeners to identify the different parts, body parts, and the different types of microbiome. Because generally, the moment we hear this word microbiome, everyone relates it to the gut. But as we now we have know that that's not the case. Your even skin, your vagina in females, different body parts, they have a different microbiome. And even when it comes to the scientific studies, we have to make sure to pinpoint from where they are picking the sample and you can inferring the results from. So that was a really, really informative information. So on that note, are there any sexual differences? I mean, are there any specific microbiome, just as you mentioned that there are vaginal microbiomes which are specific to the females? Are there any for the males, or are there any other specific microbiomes in the males and female to other specific regions which are not present in the other gender?
That's an area that's still kind of in its infancy, in part because males are more frequently studied than females, sort of like across the board. But in females, one that I have seen that comes to mind is that females may have higher levels of lactospiraceae, which I believe that's lactic acid. It could either be a lactic acid producing bacteria or a fermenter, and that produces butyrate sort of preferentially. So that one comes to mind only because I've seen it mentioned a couple times as like sort of a bad microbe, and it's not. It's just one that is producing butyrate and short chain fatty acids. So sometimes people characterize those as bad because they increase the energy harvesting from the diet.
Yeah.
That's one of the really important roles of the microbiome. So lactospiraceae is one potential. We know that there are a number of different influences on a person's individual microbiome, and there are so many that it might actually be difficult to determine what is actually related to gender, versus what's related to lifestyle,
Yeah.
pharmaceutical drug use and things like that. So that's one factor that is extremely confounding. Even, in terms of like the drinking water people are using, it's almost impossible to really control a human microbiome study because it's influenced by, anything that passes through is a potential influence. But one thing that we've gathered from rodent studies is that male and female rodents seem to have different microbial responses to ketogenic diets. So there's some clue there that perhaps there is some core microbiome that's different in males versus females, and that core microbiome might influence our entire microbiome's response to dietary changes. So that's one thing to think about. The other would be potentially the role of estrogens, mostly in postmenopausal women. So in premenopausal women, circulating estrogen doesn't seem to have any sort of correlation with a specific microbiome profile, whether that's endogenous or exogenous, like a birth control. But in postmenopausal women, there does seem to be a relationship. So those microbes are able to metabolize estrogens, and anything that they metabolize, anything that's acting as a nutrient source for them could influence their numbers and their ability to thrive. So that's another thing to potentially think about. Now, there are some other potential confounding factors. So in women, there have been relatively few studies looking at the microbiome or the fungal microbiome. But the Candida species seem to be overrepresented in females versus males. But that could be because of the proximity, but there's a close proximity between the vaginal canal and the rectum where they're collecting the fecal samples. So it could be that there's just some translocation of the Candida species. And then there are some species that could pass through just from diet or just from the oral cavity. So there's actually even debate about whether Candida are residents, like living full time in the GI tract, or if they're just passing through because we're swallowing them. And so like even oral health could play a role. And like, do males and females have like different, dental hygiene habits? So there's so many things that could potentially influence the difference in the male versus female microbiome. But it's one thing that we're actually looking at in a RCT that I'm involved with. I've got a colleague out in Tennessee who I met a few years ago at an ISSN conference. Actually, he'd done some probiotic research in athletes, and so I approached him when I was with RP and said, we might be able to get some funding and whatnot to support this study. And so we are doing a study that's looking at both male and female collegiate athletes and determining if there are any sort of baseline differences in their microbiomes and how that might be related to diet, because we're doing dietary protocols. And then we're also putting them through, or also have put them through, we're in that analysis phase now, a really rigorous, grueling resistance training experience. And then we're measuring markers of intestinal permeability and intestinal damage and getting subjective ratings of GI distress. And we're hoping that maybe we can draw some correlations between their baseline microbiome characteristics and GI distress, because one thing that we do see in the literature is that females are more likely to experience exercise-induced GI distress.
Yeah.
And it's not really, it's not really clear what the reason for that is. So hopefully we can, start to clarify that a little bit.
Definitely. That's a really, really good piece of information. And also, when I read your book, you have mentioned those genetics and epigenetics, especially the one thing which fascinated me, that example of having a vaginal delivery versus C-section delivery, right? How these two different types of delivery methods can change the microbiome of the offspring. So, would you like to throw some light on how this pans out in actual world?
Yeah, so this has been a really interesting area, in part because it's so controversial, and
Yeah.
also because there have been some kind of like upheavals of, what was commonly understood as being true at the time. So a lot of the controversy centers around when we're first colonized, whether we are colonized in utero, where it should be a sterile environment,
Yeah.
or if we are colonized, in the process of birth. So as the infant passes through the vaginal canal, they are born usually kind of like face down. And so they get a combination of the vaginal microbes and the fecal microbes in their nasal cavity, their mouth, their eyes, and that is how they're initially colonized if born vaginally. Whereas if they're born via C-section, their first exposure will be the skin microbiome, which looks very different again. So we're seeing that instead of being colonized by lactobacilli and bifidobacteria, we're seeing more like streptococcus and staphylococcus in C-section births. And so this is so that was kind of like the first controversy, like where are we actually being colonized, because some samples have shown that samples from like in utero, they have some bacterial colonization, and then other labs say that's just contamination, you didn't do that right. So that's still up in the air. So the previous sort of held belief that was pretty well received for several years was that babies born via C-section were more likely to develop obesity because of that initial colonization of microbes. But what they found later was that these studies weren't accounting for the BMI of the mother.
BMI.
Yes, so mothers with a high BMI with obesity are more likely to have a C-section birth, and then there's a strong correlation between maternal BMI and child and the BMI of the offspring. So it looks like it's probably more likely that that's the mediating relationship more so than the microbiome. But where we are seeing strong relationships that haven't been challenged yet would be the relationship between C-section birth and the development of allergies later in life. Development of allergies, either food allergies or skin allergies, so like atopic diseases and things like that. And the theory there is that because the early microbiome is so influential in shaping the infant's immune system, that initial low diversity could play a role in the development of these allergies later on, because the immune system is, for lack of a better term, uninformed and responding to things that it shouldn't respond to. Whereas, the babies that are born vaginally, they're exposed to the lactobacilli, yeast species. Yeast do appear to play an important role in immune sensitivity and these bifidobacteria. So these microbes together are helping to regulate the immune response, so it's not an overreaction, but also to keep it informed of which microbes are friendly, which microbes are potentially pathogenic. And there could also be a role of that microbiome in facilitating the sort of the closure of these tight junctions between cells. So infants they have very permeable intestines.
Yeah.
So, there's a high oxygen environment, and there's more ability for larger peptides to leak between the cells and come into contact with those immune cells in that connective tissue layer. So that's another theory, that perhaps because of that low diversity, that sort of dysbiosis, that they remain permeable for a longer period of time, and then immune cells come into contact with this peptide, they've never seen it before, and they say, oh my gosh, this must be something, to attack, and it's just, from peanuts or whatever.
Definitely. So can we say the same thing, what we have a correlation and causation between vaginal versus C-section in comparison to breastfeeding versus other means of supplying the milk to the offspring? So does that also have the possibility of changing the microbiome of the offsprings? Because you just mentioned that there is a microbiomes on the skin as well. And if a mother is not able to breastfeed that much often, will that alter the behavior of the microbiome of their offspring?
Yep, that's another potential influence. So breast milk contains microbes, but potentially from the milk ducts, antibodies, and human milk oligosaccharides. So those are specific carbohydrates that we haven't been able to replicate, in sort of like food production. So that breast milk, and the breast milk is going to be also like unique to the mother as well. So it could be potentially, that like with the diversity of the mother's diet could influence, the components of her breast milk. And, she's going to be also assisting in the development of the infant's immune system because of those antibodies. And that's not something that we have in formula either. So, of course, I fully in support of the the idea that like fed is best. Like, if you can't breastfeed for some reason, then, giving your baby whatever nutrients you can, that is of the utmost importance. And so, sometimes women just don't produce enough milk, or, there's something going on. So it's just a necessity. But fortunately, formula is improving, and, they have started to sometimes add probiotics to the formula. And, that is to, hopefully help to increase like the levels of bifidobacteria in the infant gut. So they're trying to produce, also these oligosaccharides that are similar to the human milk oligosaccharides. So, it's very similar in terms of like the macronutrient content, and macronutrients themselves can influence the composition of the microbiome, but we still are kind of missing out on, the components of human breast milk. So that's another potentially confounding factor. Early in life, like if you were at a C-section birth and then were breastfed, that at least you started off with a different set, like a different core microbiome
Yeah,
than the infant that was born vaginally and breastfed.
definitely. Really good piece of information, and it would help listeners a lot. So, with that in mind, how much role does gut health plays in optimizing our digestion? Because our targeted population, or the people who are reaching you and are reaching me for their as a whether they are lifestyle clients or strength and conditioning client, your macronutrients, micronutrients, all those diet plans or diet charts will come into the play later on. But first, we have to see that your actual engine is running in a correct manner, which is processing the entire fuel, right? If it's not processing the entire fuel in a correct manner, no matter what kind of intervention we try, whether we go higher in a surplus, in a deficit, or whether we try to fix some micronutrient deficiencies, it will be a hit or miss, or it will not be as optimized as we want to be. We want it to be, and it will dump even more noise to the signal, so it will becomes really murky to identify what is the actual signal and what is the actual noise. So what is the role of gut health when it comes to the digestion of a general population or even a athletic population?
Oh, sure. Well, the microbiome is, because of its extensive set of genes and its functionality, it does play important roles throughout the lifespan in a number of different ways for human just physiology and normal functioning. So there are theories that it plays a role in brain development, and from there appetite and feeding
Yeah,
behaviors, that it could play a role in immune function, which obviously is going to affect our training capacity. If we're sick, then we're not going to be.
recovery. Yeah,
Yeah, exactly. And in even in the adaptations to exercise, potentially because of its role in immune function. So we do actually need an immune response to adapt to exercise and get better. And it also helps to extract energy from the diet. So energy harvesting is the term that we use to refer to this. And basically what those microbes are doing is taking indigestible carbohydrates, dietary fibers, and converting them to short chain fatty acids. And the microbes are not doing this really for us. They're doing it for themselves. This is how they're gaining energy from the things that we're not using. But then by doing that, they're converting something that's not the energy is not available to us. They're making that energy available to us. And there are estimations from very limited evidence that this could increase our energy absorption by up to 200 calories a day. So some people might not, and this is a rough estimate that they're using based on like doubly labeled water and energy lost in fecal matter. So again, it could be way less than that. It could be more. And there's a wide range. They've seen like some people don't seem to harvest any additional energy from the diet. They just like there's fiber passes in, comes back out, and they haven't gained any short chain fatty acids that have been measurable. And so in that case, they may be producing more gas rather than the short chain fatty acids. And the other thing that they may be able to do is to clear lactate, or well, I shouldn't say clear lactate, that doesn't help with our performance, but they can convert lactate to glucose, or excuse me, propionate.
Pyruvate. Propionate.
Propionate. So they convert lactate to propionate. But there are other microbes that actually can also produce glucose. So they're basically performing gluconeogenesis for us. Or they are, with lactate, they're consuming that lactate, which is not a helpful, energy source for us.
So,
Yes.
as you have mentioned, all these frameworks, and we have established a lot basic frameworks and the guidelines. So, how can a particular layman person can identify that there are some kind of a mismatch happening because of that they are having some kind of issues? So, what are the, you can say, guiding factors, or what are the kind of symptoms that people should look out for when it comes to the impairment of their gut health?
Sure, yeah. So there are really no outward factors or outward symptoms of what we would term to be an imbalanced microbiome or dysbiosis, with the exception of if a person has an infection, like they have a C. difficile overgrowth, or they have a foodborne illness where they've ingested a microbe that's not a normal inhabitant, and they'll have severe vomiting and diarrhea and things like that. But when it comes to even trying to determine what's like the healthy microbiome, we haven't established that yet. So we don't actually have reference ranges for the normal inhabitants of the gut. And we don't have, like you wouldn't be able to tell, like, oh, I have too much lactobacilli or too few bifidobacteria, because I have these crazy symptoms. But there are red flags of potential diseases that people should be aware of, so that if they notice this, then they can go to the doctor. They want to see a gastroenterologist who would screen them, for disease. So if they are noticing a change in like the frequency of stool, so if they are suddenly going really frequently, if they're going in the middle of the night, or if they're feeling very constipated. So if they're going less than 3 times per week and they're straining often, then that's a sign, to go to the gastroenterologist and get checked out. Or if they're having frequent diarrhea, then that's another sign to go and get checked out. If they're noticing strange color in their stool, like it's very black, or there's bright red in it, or if it's very yellow, or if they're noticing an extremely foul smell and there seems to be oil in the water, those are signs of either blood in the stool or fat malabsorption, respectively. So keeping an eye on really strange colors. Sometimes they might see green. So if they're having diarrhea and they're seeing that it's green, that means that the microbes haven't had time to metabolize certain proteins in it. And so that means that it's gone through body very quickly. And so those are some, signs to get checked out. Or if you're noticing that your the size of the bowel movements has changed, so it's very, very narrow rather than sort of like the normal like log shape that we would see, then that could be a sign of a blockage or something strange going on that would not be as noticeable maybe. So definitely all signs to get checked out. Or if they're having severe abdominal pain, then and a lot of cramping and things like that, to go get looked at for that as well. So those are all sort of like red flags, and especially if they have a known family history of bowel disease, got a family history of colorectal cancer, celiac disease, or inflammatory bowel syndrome, or even irritable bowel syndrome, that though there could potentially be a genetic factor in there as well, and to go get that checked out. In terms of things like bloating, that can be very subjective. Sometimes people, they'll feel like they're bloated, but it's either just a normal amount of gas production, or there's not really, bloating going on. They just might feel like overly full. But if they are experiencing, stomach distension and things like that, so their stomach is like looking really stretched out and they're experiencing frequent bloating, then they can go checked out for that as well.
Definitely. So we just have to identify the green flags and the red flags, and if there is no issues, if there are no red flags, do not try to fix which isn't broken. But when I read through your book, you have mentioned that varying ranges of constipation and varying ranges of diarrhea as well. So that gives us a really good insight. So basically, when a person feels like there is some kind of a irritability in their bowel movement, some kind of frequency changes, the way they are experiencing their bowel movements or the quality of the bowel movements, that can give them brief insights on something is going wrong or something needs to be checked. So that's really, really great things to know about. Also, one thing that you have mentioned in your book that we also change our gut microbiome based on the foods that we eat, right? So, should we eat according to our gut microbiome, or is it the other way around that our microbiome adapts the way that we eat?
Well, sort of both. So our
Yeah,
microbiome is really adaptive,
adaptive.
and it will change, it will respond to our dietary habits. And this can happen very quickly in as little as 24 hours. But in terms of really significant lasting change, it's more likely our long-term dietary patterns that will really determine which microbes are long-term, sort of like long-term residents. But that being said, the whole microbiome itself is very stable. So after about the age of 3, we end up with a stable microbiome. And there's theoretically even a sort of a core human microbiome, that some percentage of the microbes are there simply because we are a human species. And we do see that, for example, when we transplant from a human donor to a rodent recipient, that not that like the human donor sample isn't maintained in that rodent. So there is a change simply due to the rodent's uh, 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 anatomy and physiology of their GI tract and their dietary patterns as well. So it's not a perfect match. It's not like it takes and then it's 100% a human microbiome, sort of a mouse flavored microbiome. And so with the human microbiome and its response to dietary patterns, if we go to extremes, it's been shown that within 24 hours, if a person were to switch to a completely zero plant, zero fiber diet and have just meat and cheese, that they would see significant changes in the relative abundance of certain microbes. So usually it's a loss of bifidobacteria. That's something that we've seen in different diets that have low fiber in common. So if we have like a low soluble fiber intake, low prebiotic fiber intake, that bifidobacteria levels will usually drop, whereas Bacteroides levels will increase because Bacteroides is more adaptive. It doesn't have as much of a requirement of just carbohydrates, so it can use proteins for energy. And so we see this sort of relative, again, we're talking about like the pie chart. So bifidobacteria goes down, Bacteroides go up. So sometimes that relative abundance is just due to the loss of one microbe, not necessarily the overgrowth of another. So that's why it can be a little bit confusing when we're looking at that versus just numbers. But we've also got data where actual numbers of bifidobacteria are falling and Bacteroides are elevated. So that's just kind of one example of a characteristic change that has been replicated with these dietary interventions that are kind of on the more extreme level. But when it comes to picking between like a prudent omnivorous diet versus a vegan and vegetarian diet, we really don't see the same significant differences. So vegans and vegetarians and omnivores don't always have significantly different microbiome profiles because they're all eating proteins, carbohydrates, and fats from different sources. Yes, but it's not so different that we're seeing like really stark contrasts. But that being said, this is assuming that the omnivorous diet or vegan or vegetarian diet is a prudent one that provides adequate fiber. So what we have seen in a recent study on bodybuilders was
Yeah.
that the bodybuilders that were eating a low fiber diet had lower microbial diversity than the bodybuilders who were eating adequate fiber. And their microbial diversity was actually the ones who had inadequate fiber. It was no different from the sedentary controls. So it could be that those microbe accessible carbohydrates and those prebiotics that are used preferentially by our beneficial microbes are mediating, our response to exercise and are necessary for a full
milieu of microbes that we would expect to see. And it's the same thing with the functionality. So when we're looking at gene expression in the microbiome, we're looking at a snapshot of what they've been up to recently. So if we're feeding, a low fiber, high protein diet, we're going to see more genes being expressed for proteolytic fermentation. And the opposite is true when we're feeding a carbohydrate, low protein diet. So these tests that, test your fecal sample and tell you which diet to use are kind of science fiction because if they're really looking at that and they're saying like, these are the genes that you're actually expressing, it would just be a snapshot of what you've been eating lately. If they're just looking at the genes that are available, but not necessarily what you've been expressing, they're just saying this is what your microbiome can possibly do, but we don't actually know what it's doing. It's like looking at a recipe
Yeah.
book. So what's more prudent, what makes more sense is to synthesize just the kind of basics of microbiology. Like, what do these microbes need to use for energy? They need carbohydrates. Where are they going to get that? From our indigestible dietary carbohydrates or from the mucus layer of our intestine, which is what we don't want them to use. So, kind of going to those basics and then applying that to intervention studies and observational studies, then we can see that it makes a great deal of sense to eat a variety of plant foods to provide a variety of these dietary carbohydrates for them. And we don't necessarily have to remove animal proteins from the diet, but just keeping in mind that proteolytic fermentation usually leads to the production of some metabolites that could be harmful or play a potential role in cancer pathogenesis. So, making sure that we have enough fiber that they're using that instead, and making sure that kind of like fits within sort of this prudent dietary pattern.
Hello, listeners. I hope you would have liked this episode of the podcast series, and if you did, please feel free to share it with your friends, families, and your colleagues so that they can also get benefit from this science-based discussion that we just had. With that in mind, this podcast, along with all the episodes, is now available on all the major platforms like Spotify, Apple Podcast, Google Podcasts, along with the audio-visual media YouTube. So, with that in mind, let me bid adieu for this time, and I will see you in the next episode.